See this cow? He really wants to get strong for the Cow Olympics, but he doesn't know how. He talks to his friend the chicken, and he says that cow can build protein cells with cell replication. To be able to build up strong muscle cells for cow soccer, you need to use your DNA to create more muscle cells. This is done through mitosis, the process of cell division. In this process, deoxyribonucleic acid, also known as DNA, the double helix amino acid made of two phosphate sugar backbones and nucleotide bases, A, G, C, and T, give the cell the instructions for division. But how do we divide our DNA when we create new cells? Well, it all starts inside the nucleus. First, to replicate your old DNA into new DNA, you need to unzip the double helix to access the bases with different enzymes, and then later relink the DNA. The enzyme that opens the DNA and unravels the helix so we can copy the DNA is called helicase. The enzyme that opens the DNA and unravels the helix so that we can copy the DNA is called helicase. The enzyme that then re-adds complementary base pairs back onto open strands of DNA to complete replication is called polymerase. Polymerase connects one strand taken from the DNA and another from free-floating base pairs inside of the nucleus. The enzyme that then finally connects back together the DNA strand and RNA strand and encloses them is called ligase. Now that you know how to replicate DNA during cell division, now you need to know how to create proteins using your newly replicated DNA. Different types of RNA are used throughout the process. RNA stands for ribonucleic acid. RNA differs from DNA in its backbone and replacement of thymine with uracil in its nitrogen bases. To be able to create new proteins, you're going to need to take reference from your original DNA strand for a brand new cell using your messenger DNA side of the newly split DNA strand. Messenger RNA is created throughout polymerase, where a DNA strand is read by the enzyme, and from it, a separate copy strand of mRNA is created. After studying the sequence of the DNA, you begin the process of creating new proteins in the ribosomes. To tell the ribosomes how to create each protein cell, a strand of DNA is fed through the ribosomes, which consist mainly of ribosome RNA. Then, the duplicated DNA strand is fed through the ribosome, which each set, or codon, of DNA is matched with a corresponding set of nitrogen bases from translation RNA, which attach to the ribosome known as the anticodon. When the two sets link, part of the protein is formed at the tail of the tRNA, which repeats then to create a full protein. The process stops when a certain pattern of nitrogen bases is fed through the ribosome and the completed protein is detached from the ribosome itself. And that is how you create proteins using your own DNA. Now go out there and win a gold.